Welcome to this first video of B2 AQA GCSE Biology. We're going to start with the first topic of B2, which is cell structure. Now, all animals and plants are made up of many cells, and because of this they're called multicellular organisms, and these cells have similar features. So in animal cells and in plant cells they have a cytoplasm, and a cytoplasm is where all the chemical reactions inside the cell take place. Animal and plant cells also share nuclei. So the nucleus of an animal cell, or the nucleus of a plant cell, has the genetic information, and it also controls the activities of the cell. They both have as well ribosomes, and ribosomes are where protein synthesis happens, so that's where protein is made. And they both have mitochondria, and mitochondria are where respiration happens, and respiration, remember, produces energy which the cell can use to then carry out lots of other chemical reactions. And then finally they both have a cell membrane, and a cell membrane controls what goes into and out of the cell. Now, plant cells also have three other features animal cells don't have. Plant cells have got chloroplasts, which can absorb light energy using it in photosynthesis to create glucose. Plant cells have a vacuole, which is where the cell sap can be stored. And then they also have a cell wall, which is for structural support, and it's made from a molecule called cellulose. Now, you also need to be aware of bacterial cells and yeast cells. So before I told you that animals and plants are multicellular organisms. Now, bacteria and yeast are unicellular organisms. This means they're made of one cell. Now, because of this, they have special features. Like, for example, they both need a cell wall because they need that structural support that you can only have if you're unicellular and you've got a cell wall. So bacteria tend to have this kind of shape, and they have a tail behind them. And the tail is called a flagella. Now, the flagella is just so the bacteria can move. And then around the outside of the bacteria, it's got a thing called a slime capsule. And the slime capsule is a protective coating. Now, just inside the slime capsule, like I said before, they have a cell wall. And the cell wall is for support, structural support. And just inside that cell wall, there would be a cell membrane, which again controls the substances moving into and out of the cell. Inside a bacteria, they've got cytoplasm, just like animal and plant cells. But unlike animal and plant cells, they don't have a nucleus. So instead, bacteria have just a single bacterial chromosome. And, quite unusually, they also have rings of DNA called plasmids. Now, yeast have a different structure again. Yeast have cytoplasm. They have a nucleus. So in this way, they're quite similar to animal cells. And they have a cell membrane, which controls what goes into and out of the cell. But one thing they do have that animal cells don't have is they also have a cell wall, which is important for them to maintain their structure. Now, cells can become specialised. If a cell becomes specialised, it's called differentiation. And if a cell differentiates, it can't then change back to how it was before. So if you have an animal cell called a stem cell is a cell that's got the potential to change into any other cell. If it changes, it might change into, let's say, a sperm cell. Now, a sperm cell is a specialised cell because it's specialised to perform a particular function. Its function is to try and get to the egg. Now, if we look at this sperm cell, some of the specialised features of it is it's got loads and loads of mitochondria in its midsection, so mitochondria. Now, in an exam, they might point out this is a specialised cell called a sperm cell. It's got lots of mitochondria. Why is this a beneficial adaptation? So why has it become adapted in this way? And you could say, well, mitochondria, that means there's going to be plenty of respiration. So lots of energy is going to be released, and the sperm is going to be able to swim. Another adaptation you might be asked to pick out for this specialised cell is the fact that it's got a tail so it can swim. So if you're given the structure of a particular specialised cell, don't freak out and think, oh, I've never come across that before. Just think logically, well, what adaptations does it have? How does that? So now we're going to move on to the second topic in B2, and this is diffusion. Now, diffusion is the net movement, and this means overall, so net movement or overall movement of particles from where they're at a high concentration to where they're at a low concentration. So we're going to look at a few examples now, and we're going to talk about what's important for diffusion to occur. So here we've got some dye molecules being dropped into some water. Now in this area here, the dye molecules are at a high concentration. However, after some diffusion has taken place, the particles have spread out and they've reached equilibrium. This is where there seems to be an even distribution of the particles throughout the liquid. Now, diffusion only happens in fluids, so liquids or gases, and it's where these particles move from that high concentration to where they're at a low concentration. Now, you can also say it's where they move down a concentration gradient. So a concentration gradient is formed when you have a high concentration, or an area where there's a high concentration, and areas where there's a lower concentration, and the particles will naturally want to move from a high to a low or down the concentration gradient. Now here's two examples of where diffusion happens in the body. The first example is inside the villi of the small intestine. So these villi are folds in the wall of the small intestine. And what happens is when a piece of food comes along to the small intestine, the food will diffuse into the villi. And the reason it's diffusing into the villi is because the villi are packed full of capillaries. Now, these villi are well suited to diffusion because, number one, they are just one cell thick. This means the diffusion pathway is short. The other way they're well adapted to this diffusion is the fact that these folds increase the surface area. So this means more diffusion can take place. And then finally, the last way they're really well adapted is the fact they've continually got blood flowing through them, because inside the villi there are loads and loads of capillaries, and these capillaries are bringing fresh blood into the walls of the intestine and taking away any blood that is full of like this absorbed food. So because of this, the absorbed food, or the food that's going to be absorbed inside the small intestine, is constantly coming into contact with fresh blood that doesn't contain huge amounts of dissolved food already. So there's a concentration gradient. So I'm going to put here, fresh blood maintains a high concentration gradient. Because the food is always going to want to move from where it's at a high concentration inside the small intestine to where it's at a lower concentration inside the fresh blood. Now, the second diffusion example I want you to look at is the alveoli inside the lungs. So the alveoli are where gas exchange takes place, and the alveoli are small pockets inside the lungs that get filled with air. And around the outside of every alveoli there's lots of capillaries. So here you can see this is a capillary around the outside of an alveoli. Now, just like before in the small intestine, this capillary has an important job. It picks up materials from inside the alveoli. So if you imagine air comes into the alveoli, and that air is going to be rich in oxygen. And when it gets inside the alveoli, it comes into contact with these capillaries which are next to the walls of the alveoli. And these capillaries have got continually fresh blood coming into them. And this blood is deoxygenated blood, so it's low in oxygen. Now, when the oxygen comes into the alveoli, it's going to want to move from the alveoli into the capillaries, because the capillaries are full of deoxygenated blood. So if I put here, deoxygenated blood creates a high, or I should say a steep, concentration gradient. Grad. So deoxygenated blood creates a steep concentration gradient. Another way that this is well adapted to diffusion is the fact that these walls of the capillary are one cell thick again, so we've got a really short diffusion pathway. Then finally, the alveoli themselves are extremely well adapted to diffusion because there are many alveoli. So I'm going to put here many alveoli create a large surface area. So overall, diffusion is a net movement of particles from a high concentration to where they're at a low concentration. In order to increase the rate of diffusion, you can increase the surface area, you could shorten the diffusion pathway, or you can increase the concentration. So now I'm going to move on to proteins. Proteins are long chains of amino acids. So if you look at this sequence bit here, this is a long chain of amino acids that are all joined together. And eventually, once these are all joined together, they fold in a particular way to form a structure like the one we see here, and this is protein. Now, proteins all have these specific structures, and this can be really useful. So proteins have many uses in the body. Number one, they can be used as structural components. And what we mean by structural components is muscles. And you always say protein is used for growth and repair, so that makes sense. It's muscles. A second use of protein is hormones. So hormones are chemicals that travel through the blood and signal various things to the body. They can also be used as antibodies. And remember, antibodies have specific shapes that fit to specific antigens on the surface of pathogens, and then they kill the pathogens. So again, this specific structure of protein is important. And then number 4, they can be used as catalysts. Now, catalysts are chemicals that speed up the rate of reaction without being involved or being used up in the reaction themselves. And biological catalysts are called enzymes. So these are 4. So enzymes are biological catalysts. They speed up the rate of reactions that happen inside your body, and they're incredibly useful. Now, often scientists talk about enzymes like locks and keys. This is because enzymes have really specific shapes, and they have specific areas called the active site, and these active sites will only fit one substrate. So for that reason, they're a bit like locks and keys. The substrate is the molecule that sits inside the active site, and only one substrate fits one lock or one enzyme. Now, when the two meet together and the enzyme has a substrate in its active site, it's called an enzyme substrate complex. So the model refers to this as the lock and key complex. At this point, the lock has fit into the key. Now, when you turn a key in a lock, something happens, and it's the same with an enzyme. Once a complex is formed and a substrate has fit inside an enzyme, something will happen to that substrate. Either that substrate will be broken down, or that substrate will be joined to something else. Now, enzymes that use in the digestive system break things down. So let's say this substrate was starch. Starch would come along and sit in the active site of the enzyme amylase, and then it would be broken down into 2 glucose molecules, which would then be released. Now, enzymes are also useful not just inside your body, but also in industry and in the home. Now, in industry, enzymes can be used to create slimming foods, and the enzyme that is used for this is isomerase. Now, isomerase converts glucose into fructose. Now, fructose is much sweeter than glucose, so if you use the same amount of fructose as you would glucose in something, it would make it incredibly sweet. So what this means is, if you create a slimming food and you want it to have less calories, you can use fructose to create just as much sweetness as glucose would, but you'd have to use less fructose. So overall, you're going to use less calories. So lower amounts of fructose can be used to make the same sweetness as glucose. Another way it can be used in industry is you can use proteases to break down protein in baby foods. Advantage of this, the baby isn't going to choke on the food, and yet they'll still have access to loads of protein in the diet. Now, disadvantages of using enzymes in industry, often they need really specific temperatures to work, and also they can cost a bit more. Now, in the home, enzymes can be used in biological detergents. So this means washing powders. So let's say you're eating your spaghetti and you get it all down you. If you use biological detergent in your wash, it might contain proteases, it might contain lipases, and these can break down the fats and the protein inside that food that you've got on your top and then wash it away. Now, biological detergents, there are some positives and there are some negatives. The negatives might be, you might be allergic to them, and also they can pollute water courses. So if you're using it in your washing machine, then your washing machine runs off and the runoff is in near a stream, etc., it might pollute them. But the advantages are you can wash at lower temperatures. So it saves energy and it saves your electricity. Now, enzymes are used throughout your digestive system, and you need to be aware of all the parts of your digestive system, what they do and what enzymes they release. So we're going to start off in the mouth. Now, I haven't really got a mouth on this diagram, so I'm going to draw one in. Now, inside your mouth, your food is ground up using your teeth. So this is the first part of digestion. And also, saliva from your salivary glands is added to the food. So I'm going to put here salivary glands. Now, the salivary glands produce an enzyme called amylase. And amylase breaks down starch into glucose. So amylase is a type of carbohydrate. It breaks down the carbohydrate starch. I'll put here carbohydrates. Now, anything that ends in ase is going to be an enzyme. Now, once your food has been squirted with saliva and it's been chewed up, it starts to go down the esophagus, and this is the tube between your mouth and your stomach. Then once in your stomach, your food comes across two things. It comes across acid, and it's hydrochloric acid, and it comes across the enzyme protease, because your stomach makes proteases. Now, proteases break down proteins into amino acids, and the hydrochloric acid kills any bacteria that might be on your food. Just try and stop you getting food poisoning. And also it creates an optimum pH for the proteases to work, because proteases like working in low pHs, or that's where they work best. And if you increase the pH, you could end up denaturing them. So your stomach is actually made of 3 different tissue types, and you also need to know this. So the 3 different tissue types that your stomach are made up of. First layer is the epithelial layer, and this is the layer that covers the inside and outside of the stomach. Then you've got the muscular layer, and that churns the food. And then you've got glandular layer. And the glandular tissue layer releases enzymes. And anywhere you've got glandular tissues inside your body, they're either going to release an enzyme or they're going to release hormones. So your stomach has those 3 different layers, and they're dead important. Now, moving on from the stomach, your food starts to go into the small intestine. And inside your small intestine, this is where your food is going to be absorbed. But before it can be absorbed, it needs to be broken down a little bit more. Now, in order to break down your food a little bit more, your liver and your pancreas secrete different substances onto the food as it's just about to enter the small intestine. So if you imagine, the pancreas looks a bit like this behind the stomach, and here what you can't see is the tube where the stomach meets the small intestine. There are ducts or little tubes that run from the pancreas and run from the liver, and can secrete chemicals onto your food. So the liver secretes a chemical called bile. And bile has two important functions. Number one, it emulsifies fats. And that means it breaks up fats from being quite large molecules into much smaller ones. And this increases the surface area. And then the other thing bile does is it neutralises the stomach acid. And this means it doesn't damage your small intestine as the food travels through covered in acid. And it also means it creates an optimum pH, an optimum slightly alkaline pH for lipase. Now, pancreas secretes lots of things. The pancreas secretes 3 different enzymes. Number one, it makes and secretes amylase, and you know what amylase is for because that's at the top of the page. It breaks down starch into glucose. The pancreas also secretes proteases, and that's also just above this, so you should know what protease is for. It's for breaking down proteins into amino acids. And then the last thing it secretes is lipase. And lipase breaks down fats into fatty acids and glycerol. Now, inside the small intestine, this means all of these 3 enzymes are released. So amylase, protease and lipase are all released in the small intestine. They're all made as well in the small intestine, and they break down the food so it can be absorbed. Now, the final stage in the digestive system is the large intestine. Now, in the large intestine, any water that can be reabsorbed will be reabsorbed, and faeces will produce. This faeces will then be stored in the rectum before being released through the anus. Now we're going to move on to photosynthesis. Photosynthesis is the process plants carry out to make their own food, and photosynthesis happens in chloroplasts inside plant cells, and inside the chloroplasts they contain a pigment called chlorophyll, and chlorophyll is the thing that absorbs the sunlight to carry out photosynthesis. So the word equation for photosynthesis is carbon dioxide, which the plant gets from the atmosphere, plus water, which the plant gets from the soil, goes to glucose plus oxygen. Now, the similar equation for this is 6 CO2 plus 6 H2O goes to C6H12O6 plus 6 O2. Now, photosynthesis is really important because it produces this glucose, which the plant uses for many reasons. So the plant uses glucose in several different ways. Number one, it uses it in respiration, so that it can create energy for various chemical reactions, or I shouldn't say create energy, it should transfer energy. Number two, it's used to create fats and oils for storage. Number three, it's used to create cellulose, which strengthens cell walls and makes cell walls. And then number four, it's used to create proteins. And obviously protein is dead important because protein is used even in enzymes, and you need enzymes to carry out photosynthesis. Now, it's worth noting here, to make proteins, plants also need to absorb nitrate ions from the soil. Now, plant leaves are adapted to maximise photosynthesis, so they have different layers to do different jobs. and you need to know about the structure of a leaf. So here's the internal structure of a leaf, and at the top we've got this thing called a waxy cuticle, and it's also at the bottom. The waxy cuticle tries to prevent water loss. Just underneath that you've got the epidermis, and the epidermis is just the covering layer. It covers the outside of the leaf. Just inside of that you've got the mesophyll layer, and this is broken into two parts. The palisade mesophyll, which is where photosynthesis happens, and these cells are positioned at the top of the plant so they can get as much light as possible, and they're full of chloroplasts which have chlorophyll to absorb the sunlight. And then the spongy mesophyll has got air spaces in between those cells, and that's where gas exchange happens. And gases diffuse through holes in the underside of the leaf called stomata, and either side of the stomata are guard cells which can open and close to let more or less gas in. Now, one structure that isn't shown in this diagram and sometimes is shown as a large circle with different sections in it is the vein. Now, the vein of the plant contains two different types of tissue which are important. It's got the xylem and it's got the phloem, and these are both two types of tissue that are shaped into tubes that run up the entire length of the plant. And the xylem carries water around the plant, and the phloem carries sugars. Now, you need to know about the limiting factors that can impact on photosynthesis. So a limiting factor is one that determines the rate of photosynthesis when all other factors are readily available. So these three factors are light intensity, carbon dioxide and temperature. And often these graphs come up and you need to be able to describe what they show. So I'm going to talk through this now. Now, light intensity is a limiting factor of photosynthesis because as you increase light intensity, the rate of photosynthesis increases. However, there comes a point where no matter how much light you add into this reaction, the rate of photosynthesis just stays the same. And this is because at this point, light is no longer a limiting factor. Another limiting factor must be limiting photosynthesis. So here you would say light is no longer a limiting factor, and you'd probably give an example for what could be. So for example, at this point CO2 could be the limiting factor, or maybe temperature. Now, carbon dioxide is similar. To begin with, at low levels carbon dioxide is a limiting factor because as you increase carbon dioxide the rate of photosynthesis increases. However, there comes to a point where no matter how much more carbon dioxide you add into the greenhouse, the rate of photosynthesis stays the same. So here, carbon dioxide is no longer a limiting factor. It's no longer determining the rate of photosynthesis. Something else must be, perhaps light or temperature. Now, the temperature graph is slightly different because the temperature graph takes into account denaturing of proteins. So imagine here you've got temperature as a limiting factor because you can tell, because as temperature increases, so does the rate of photosynthesis. However, it gets to a point where no matter how much you increase the temperature, the rate of photosynthesis doesn't increase anymore. In fact, it decreases. So the reason that it decreases here is because enzymes are being denatured. So if you put an enzyme in a pH that's not correct, or you heat it up so it doesn't work properly anymore because it's too hot for that enzyme, what happens to the enzyme is it denatures and it stops working. Now, what this means is if you imagine this was the shape of the enzyme to begin with, and it had a really specific active site in which only one type of substrate would fit in. If you heat up this enzyme so much that all the bonds in it start to break, the shape of the enzyme changes and it no longer has that specific active site in which its substrate fits. And so the enzyme doesn't work anymore, and this is called denaturing an enzyme. So with the rate of photosynthesis and temperature graph, temperature can be a limiting factor of photosynthesis at low levels, and if you increase the temperature, you do increase the rate of photosynthesis. But if your temperature gets too high, you can denature your enzymes, and so photosynthesis. So moving on to the next topic, we're going to look at sampling. And sampling is all about finding out how organisms are distributed, so how living things are placed in their habitats. Now, the distribution of organisms can be dependent on lots of different factors. For example, the temperature of the space, the nutrient availability, the light, water, and oxygen and O2 concentrations. CO2 concentrations. So let's think about plants. If you're a plant, you're probably going to want to live somewhere where you've got lots of CO2 for photosynthesis, lots of water for photosynthesis, and lots of light for photosynthesis. Whereas if you're a fish, you're going to look for somewhere where you've got lots of dissolved oxygen and lots of nutrients. So distribution can be affected by lots of different factors. If you want to look at the distribution of different organisms, you'll either use quadrats or you'll be using transects. So quadrats I would use if you're looking at separate areas. So if you want to compare the daisies in two different fields and you want to see if fertiliser has affected one of the fields, you would use quadrats. However, if you want to sample along a line, so let's say you want to sample through a woodland habitat, or you want to sample from a beach all the way up to where you go through like the shrubs at the back end of the beach and then into some trees, then that's a longer line, so you're going to use transects. For both of these, there are certain steps that I would follow each time I was answering a question on them, because commonly you're given a scenario and they'll say, how would you use quadrats to sample this area, or how would you use a transect? So if you're going to use a quadrats method, the first thing I would suggest you doing is you mentioning that you're going to create a numbered grid. Now, what this means is you're going to take your area like we have here, and you're going to split it into small sections, and you're going to number those sections. If you look here, you've got coordinates. Then what I'd say is you would use a random number generator to select grid squares. So for example, my number generator might say I'm selecting 6B, so I'd choose to sample in this area. It might say I've got a sample in F2, in which case I'd have to sample in this area. The next thing I'd say I'd use is lay your quadrat in the selected squares. So a quadrat is a metre by metre square, and you just place it in the section that's been chosen by your random number generator, and you count the number of species inside your quadrat. So I'm going to put here, count organisms. Now, depending on the question, you might have to count daisies, you might have to count daffodils, you might have to count grasshoppers. So you change this step number 4 depending on what you're counting. So you count organisms inside your quadrat. The next step I would say is repeat. I'm going to say more than 10 times. Okay? And you're going to do this randomly. Now, what you can do then is, using those values that you found, you can find the mean number of organisms in that habitat. So you might find, if you've done 10 quadrats, let's say, well, you've got 10 different samples at that point, you need to add them up and you need to divide by 10, and that'll give you the mean number of organisms per quadrat. Now, if you're using a transect, you have to start a bit differently. The first thing you have to mention is lay out a tape measure. Now this tape needs to be as long as is suitable for the habitat. So let's say we've got this habitat here and this distance is 10 metres. Well, that's fine, lay out a 10 metre tape measure. But if that's 100 metres, you're going to have to lay out something close to 100 metres instead. And so you lay out the tape measure, and I want you to put your value in for how many metres you lay it out by. Number 2, you lay your quadrat then you tell me how many metres apart you're going to lay each quadrat. Because when you sample with a transect, you don't sample randomly anymore, you sample systematically, which means every certain unit of distance, you lay out a quadrat and you count how many organisms are inside it. So let's say this was 100 metres, you might choose to lay out a quadrat every 10 metres. Equally, if this distance was just 10 metres, you might want to lay them out every 1 metre. So you choose what's sensible for your question. So number 2, lay your quadrat out every so many metres. Number 3, count the organisms that are inside your quadrat. Then number 4, repeat by laying out another 10 random transects. Now at this point we're going from systematic to a bit of random, because they can be laid out randomly anywhere along here. So I might choose to do one transect along number 4, but then my next one might be number 7 and my next one might be number 1. So that bit can be random, but the actual sampling along the transect needs to be systematic, so it needs to be evenly spaced. So that's how you sample with quadrats, and that's how you sample with transects. If they ask you how can you improve your accuracy, you could say I will sample more, so I would use more quadrats. That usually gets you the mark for increasing accuracy. Now another thing you might be asked to look at is they might say use percentage cover to estimate how much of this area is covered in grass. So for example, let's say this is a quadrat and you've got these areas like this which are grass. If they ask you to estimate using percentage cover, they want a rough estimate of what percent of that ground is covered in a particular thing. So in this case it might be, I'd say that's just under about, so that's about 45% grass. So that would be using percentage cover. And the benefits of using percentage cover are, well, if you've got something like grass, it's really hard to distinguish individual pieces of grass, so it's probably easier to use percentage cover. So now we look at respiration. Respiration is used to transfer energy, and it's really useful because the energy can be used in several different ways. So if we think about energy and its uses, the energy can be used to build larger molecules from small ones. In animals it can be used to contract their muscles. In plants it can be used to build proteins. In animals it can be used to control their temperature. And so overall it's a pretty important process, and it happens in all cells. So I've put here all cells. And the area that it happens in is the mitochondria. Now if we compare aerobic and anaerobic respiration, and we'll do this in humans, aerobic respiration happens when there's a plentiful supply of oxygen. So if you've got loads of O2, aerobic respiration will happen. It's a continuous process, and it happens all the time in plants and animals. So continuous process. Now the word equation for aerobic respiration is oxygen plus glucose goes to carbon dioxide plus water vapour. And the symbol equation is 6O2 plus C6H12O6 goes to 6CO2 plus 6H2O. So it's the exact opposite of photosynthesis. Now, for this reaction to happen, it uses enzymes. And that's important, because if something gets too hot or if the pH isn't correct, then the enzymes could stop working and aerobic respiration could stop working. Now, anaerobic respiration is where there's not enough oxygen for aerobic respiration to take place. So instead, anaerobic respiration takes place. Now, because there's not enough oxygen, the equation for this is just glucose goes to lactic acid. And this is called the incomplete breakdown of glucose. Now this process produces enough energy to get by, and it's quite a lot of energy actually. It's a sharp shock of energy. However, long term, it really doesn't produce that much energy. So if you compare aerobic respiration to anaerobic respiration, way more energy is produced via aerobic respiration because it can happen continuously and large amounts of energy can be created. Anaerobic respiration is enough for a short, sharp hit of energy, but then afterwards you've really got to start getting enough oxygen to carry out aerobic respiration. It can't last for long. So anaerobic respiration results in a thing called an oxygen debt. And an oxygen debt is where your muscles haven't got enough enough oxygen to break down glucose completely. So, like I said, you end up with this lactic acid being built up. Now, lactic acid, if there's enough of it building up, can cause muscle fatigue. And this can mean your muscles get to a point where they can't contract anymore. And this is all because of this oxygen debt. Now, because of this, when you exercise, you might find if you do something like a sprint, you get really out of breath. And after you finish your exercise, you have to keep breathing really, really heavily. And this is to repay your oxygen debt. So in order to repay your oxygen debt, your breathing rate usually increases. And so does your heart rate. And this is to try and get as much oxygen as possible to any muscles that have ended up with an oxygen debt. And then we can repay your oxygen debt, and as soon as the oxygen floods into the cells, the oxygen can be used to break down the lactic acid completely, and therefore you can get rid of the lactic acid and everything can go back to normal. Now we're going to start the genetics portion of B2. So we'll start off by looking at a body cell. Now, inside all of your body cells, you have a nucleus, and the nucleus contains your genetic information. Now, inside your nucleus, because you're a human, you've got 23 pairs of chromosomes, and you've inherited 23 of these from your mum and 23 of these from your dad. So you've got a combination of their genes. Because of this, because of sexual reproduction, you vary from your parents. You're a combination of both their genes, and that's one advantage of sexual reproduction, that you get variation. Now, if you take a chromosome, a single chromosome, and you unwind it, you'll see that it's made of DNA, and this DNA has a particular structure. It's called a double helix. Now, along the double helix, there are things called bases, and these bases code for amino acids, and amino acids then make up proteins. Now, a section of DNA that codes for a particular characteristic is called a gene. So this might be the gene for eye colour. So this section of DNA codes for particular amino acids which make the proteins which eventually are shown as your eye colour. Now, eye colour has lots of different variations, so it might be that you've got blue eyes, it might be that you've got brown eyes, it might be that you've got green eyes. Each of these different versions of the same gene are called alleles. And some alleles are dominant and some alleles are recessive. If you have a dominant allele, it will always be shown. And if you have a recessive allele, it will only be shown if you haven't inherited a dominant one. Now, your DNA is completely unique to you. Like I said, we vary from all the people in the world that we know, and it's because we've got this unique DNA that we've inherited from our parents. It's a unique combination of both their DNAs. Now, the only people who share DNA are twins. They have exactly the same DNA. Now, one of these pairs of chromosomes you need to know is called the sex pair of chromosomes, so sex chromosomes. And sex chromosomes are the chromosomes that decide whether you're a boy or a girl. If you're a girl, you'll have 2 X chromosomes, and if you're a boy, you'll have an X and a Y chromosome. Now, at one time or another, all the cells in your body will have to go through a process of cell division, and they'll either go through meiosis or mitosis. Now, mitosis occurs in body cells, and it's for growth and repair. And meiosis only occurs in reproductive organs, because all that meiosis is for is to create gametes. So if we start off with the process of mitosis, the first stage in mitosis is the DNA in your cell is replicated. So you'll go from having 46 chromosomes to all of a sudden having 92. So you've got 92 chromosomes. The next stage is the nucleus inside your cell divides. And this means you've got 46 chromosomes in each of those nuclei. And then eventually the whole cell divides, and you end up with two body cells that are identical copies to the first body cell because they contain exactly the same genetic information. So I'm going to put here nuclei split, cell splits, or I'll put cell divides. So with mitosis, you have one cell division, and you have two new daughter cells created. And they are exact clones of the first cell. So let's say you need to create more blood cells, then your blood cells will go through mitosis to create identical copies of those cells that are needed by the body, and this happens in body cells. Now, one way of remembering that is it happens in my toesies, so my body cells that are in my toes, but it happens everywhere else too. Now, the other form of cell division, meiosis, like I said, only occurs in reproductive organs. So your ovaries and your testes, and it's purely to produce gametes. So meiosis goes through the same stages as mitosis to start off with. So first of all, your DNA is replicated. So you go from having 46 chromosomes in your nucleus to having whopping 92. The next step is the nuclei split, so it's pretty much the same again as mitosis. So you have two nuclei that are splitting to form two nuclei with 46 chromosomes in each. Next step is the cell divides. And here you've then got two new daughter cells created, each with 46 chromosomes. Now here's where mitosis differs from meiosis. So meiosis goes on to have another cell division. So I'm going to put here number 4, nuclei split again. This time the nuclei haven't replicated the DNA though. So you're going to end up with two nuclei, each with 23 chromosomes in them. And then your final step is your cell is going to divide again. Now in this case, you're going to end up with 4 daughter cells. And each of those daughter cells is going to have 23 chromosomes in it. And those chromosomes will have been randomly selected. They'll have been randomly assorted. So this is another reason we get loads of variation through sexual reproduction. So the main part of meiosis, or the main feature of meiosis you need to remember, is there are 2 cell divisions, and it creates 4 new non-identical daughter cells. And those cells are gametes. So let's say this happened in the lady's ovary, then these cells created would be egg cells, but equally, if this happened in the man's testes, then these cells would all be sperm cells. So once you've created your gametes using meiosis, you'll have a mother that's created egg cells with 23 chromosomes inside them, and you'll have a father that's created sperm cells with 23 chromosomes inside them. When they combine and they fuse during sexual reproduction, this process is called fertilisation. So if you ask what fertilisation is, it's when two gametes fuse and the DNA mixes. At this point, they create a fertilised egg cell, and the fertilised egg cell contains 46 chromosomes, and this is the first cell inside the newborn's body. Now, this cell has to become every other cell in the entire body, and so it's called a stem cell because it's got the potential to become any cell in the baby's body. Now, that stem cell begins to divide, and the way it's going to divide is mitosis because it's now a body cell and it wants to create identical clones of itself. Now, once you've got a group of identical cells like this, you've got yourself an embryo, and embryos contain stem cells. Now, stem cells, like I said a minute ago, are cells which are unspecialised, or you could say they are undifferentiated, which means they don't have a role yet, they haven't specialised, and they can make any cell in the entire body. Now, obviously, this is important for embryos because they're going to go on to create a baby. However, it can also be important to scientists because stem cells we're doing lots of research on at the minute could be incredibly useful for curing quite a few diseases. So there are positives to stem cells, and these are things like maybe we could cure paralysis. If you think about paralysis, it's where somebody's been paralysed because of a spinal injury. So maybe the spinal cord has been severed while it's been broken. Now, they're paralysed because the nerve cells in the spine have been damaged. But if we use stem cells and we grow nerve cells from them, we could potentially cure things like paralysis. Equally, it could be used for leukemia. Leukemia is a problem, a cancer that affects blood cells. Well, if we can use stem cells to grow fresh blood cells, we could maybe cure leukemia. So there's loads of different possibilities for using stem cells, and one positive might be we could cure paralysis. And I've put here nerve cells. However, there are some negatives to using stem cells. The negatives are lots of people feel that using embryos is not moral because the embryos themselves obviously have no choice. They do not consent to being used. Usually the embryos are either used from abortions, or it might be from stockpiles of embryos that have been made up during IVF and then just not used. So people see it as not being moral, and they see it as a potential life that you're then ending to do your research. There is another way around using embryos though. So maybe instead of using embryos, we could use instead adult bone marrow, because the one place in an adult that doesn't contain specialised cells and still contains stem cells is adult bone marrow. So at the minute, lots more research is going on to try and look at using stem cells. So we've looked at how gametes are created and we've looked at what happens during fertilisation. Now I want you to consider somebody who's pregnant and thinking about what genes their child might inherit. Now, if it's a case of, I just want to know if they've got blonde hair or blue eyes, scientists and doctors are unable to, at this point in time, justify helping them because it doesn't matter if your baby's going to have blonde hair or blue eyes, who cares? But if you've got a genetic disorder that runs through your family, scientists can help you out, and they can give you an idea of the likelihood of your child being born with that genetic disorder. And they can do this using family trees and monohybrid crosses or Punnett squares. And both of these particular systems can give you a good idea of the likelihood of your child also being born with a genetic disorder that runs in your family. And so it can help you prepare for things like if they're going to be born with a disorder that you need to have specialist equipment for, or you might even consider a termination if the disorder is going to be that severe. Now, there are a few key words you need to know about before we get started at looking at monohybrid crosses and family trees. And the key words are here: dominant allele, recessive allele, homozygous and heterozygous. Now, I want you to consider that when fertilisation occurs, 23 chromosomes from the mother join with 23 chromosomes from the father. And each of these chromosomes is a matching pair. So, for example, from your mother, you might inherit chromosome 1, and from your father, you're going to inherit a chromosome 1. Now, they're going to contain the same genes. So, chromosome 1, if it controls, let's say, eye colour, then let's say this section here of that chromosome codes for eye colour on the mother, then you're going to have a matching chromosome over here with a matching gene coding for eye colour from the father. Now, this isn't to say you'll inherit the same gene from both your mum and your dad. You don't. You inherit two different alleles. Now, these alleles, I say different, well, they could be different, but equally they could be the same. But either way, you inherit two copies, you inherit two alleles, one from your mother, one from your father, for each gene that makes up you. So, you could inherit a dominant allele. A dominant allele is always expressed, so it's always shown. So let's say, or always shows, let's say this allele here from your mother is for blue eyes and it's dominant, and this allele from your dad is for green eyes and it's recessive. Well, if that's the case, then you'll inherit this allele here, the dominant allele. So dominant allele always shows, or is always expressed. Now, recessive allele only shows if the dominant allele isn't present. So let's change this up here, and let's say from your mother you inherited a green recessive allele, and from your father you inherited a green recessive allele. In that case, you'd get green eyes. However, the minute one of these changed, so if your father actually gave you brown-eyed allele, or if your mother actually gave you the brown-eyed allele, then that will be shown because that will be the dominant. In the exam questions, they'll always tell you which is the dominant, which is a recessive allele, but they won't tell you what that means. So dominant is always expressed, recessive allele is only shown if the dominant allele isn't present. Now, homozygous individuals are individuals that inherit the same alleles of a gene. So in this case, if we had the mother given up her recessive green allele and the father had given his recessive green allele, then it means the person created, or the baby created, would be a homozygous individual for that particular allele. And in fact, they'd be homozygous recessive because they've inherited two recessive copies of that allele. Now, if someone is heterozygous for a gene, it means they've inherited different alleles. So let's say the mother gave them a dominant brown allele and the father gave them recessive green allele. In that case, they've inherited different alleles, so they are heterozygous for that particular gene. Now, we can use these key words and in order to help us try and construct a Punnett square. Now, I'm going to show you this question here. I'm going to read it to you. Construct a monohybrid cross to determine the likelihood of a child having cystic fibrosis if both parents are carriers. Now, cystic fibrosis is a recessive disorder. Now, because it's recessive, this means you need to inherit two recessive alleles in order for you to get cystic fibrosis. So if we've got both parents being carriers, a carrier is someone who has the disease without actually knowing that they have it. So they have a dominant allele, which is for no cystic fibrosis, and they also carry a recessive allele. And the recessive allele, if you've got two copies of it present, would mean you would have cystic fibrosis. Now, if both persons are carriers, this means they must be heterozygous for cystic fibrosis because they've inherited one dominant allele and one recessive allele. Now, if you're constructing a monohybrid cross, you've got to take into account both parents and what would happen if you mix both of their gametes. Now, a monohybrid cross, another word for it is a Punnett square. If both parents are carriers, I always put the father on top here, so if I put dad there, the dad could give you sperm cells that contain a big C, in which case that would be coded for no CF. Or the dad could give you a sperm cell that contains little c. Equally, when the mum makes her gametes, she could have eggs that have the big C in them. Or equally, half her eggs are going to contain little c because she's a carrier. Now, if this egg meets this sperm, then you're going to create an individual that's got 2 big C's. If this sperm meets this egg, then you're going to have a person who's got a big C and a little c. If you've got this sperm and this egg, you're going to have big C, little c. And if you've got this sperm and this egg, you're going to have little c, little c. Now, usually, you get 1 mark if you get the correct gametes of the parents, and you get another mark if you get the correct outcomes of individuals. So there are 4 possible outcomes for this family, and they're all listed here. Now, these are known as the gametes. So these are the genes that make up that individual. Sorry, not gametes, genotype. That's what I was about to say. So these are the genotypes. These are the genes of the individual. So if we look at trying to list the genotypes, here we've got a homozygous dominant individual. Now, because there's 4 squares in total, that means you've got a 25% chance, because one of those squares is homozygous dominant, 25% of having a homozygous dominant individual from those parents. Now, these other 2 squares, so this one here and this one here, contain heterozygous individuals. The genotype is heterozygous. Now, if there's 2 squares out of 4, that means there's a 50% chance of that individual being heterozygous. Now, the final individual, there's a 25% chance of having a homozygous recessive individual. Now, you usually get another mark if you get these genotypes correct. And then your final mark is for answering the question usually and relating it to a phenotype. So these here are the genotypes, but the phenotype is the physical characteristic. So if I put here physical, what does this actually mean? And this is the phenotype. And usually it's relating it back to the question at this stage. So the question says, construct a monohybrid cross to determine the likelihood of a child having cystic fibrosis. Right, so the physical phenotype I'm looking for is cystic fibrosis. Now, the only one who's going to have cystic fibrosis is this individual here, because they're going to be the homozygous recessive. If you remember, cystic fibrosis is a recessive disorder. It only occurs in people who've only got recessive alleles present. So the final statement you usually get a mark for is for answering your question relating it to phenotype. So in this case, I'd say there's a 25% chance of having a child with cystic fibrosis. Now, this might seem like a lot of effort to go to work out if your child, or work out the chance your child is going to be born with cystic fibrosis. But cystic fibrosis is a really harmful disorder, and it can cause eventually death, and it shortens your life, and it's a horrible disorder where your lungs fill with mucus, your airways fill with mucus, and you can't stop coughing, and it's quite uncomfortable for people who have it, and they have to go through lots of treatments throughout their lifetime, and often you end up having to go through lung transplants, and it can be quite a horrible time waiting for a lung transplant, and often many people don't get it. So it might seem like quite a lot of effort to work out if your child is going to get cystic fibrosis, but actually it's kind of worth it because of the disorder and because of the problems it causes. Now, other genetic disorders that you need to learn about, one of them, which is polydactyly, are caused by dominant alleles. So polydactyly is caused by dominant allele. This means if both your parents are homozygous dominant, you are definitely going to get polydactyly. And polydactyly is where you've got extra digits. So it might mean you get an extra finger, or you might get some extra toes. Now, polydactyly, lots of people like to know if their children are going to get it or not. and they might go through some of this genetic screening that you've just seen, so with the monohybrid cross. However, it's seen as less important than knowing about genetic disorders such as cystic fibrosis. So polydactyly, many people might not go through the process of trying to find out if their child has polydactyly, and lots of people might argue that they shouldn't go through this process because does it matter anyway? It's only an extra digit, and they shouldn't think about, they shouldn't be so, what's the word, so bothered about something that's quite superficial. So sometimes when you're looking at the pros and cons of genetic screening, you can talk about the fact that if it's a disorder where it's just very superficial, is there any point to go through genetic screening, and do you think it's right that we go through genetic screening? Now, the last thing that we could do as part of a genetic screening program is you can do this. You can work with family trees. Now, family trees are quite good because they give you an overview of how a disorder has been passed through a family, and they can show you who's affected with the disorder. So usually the affected people are coloured in, and it can also show you unaffected individuals and whether you're dealing with females and males. Now they're fairly straightforward to read and to work through, so if I just give you a few pointers, if we look at the second generation, we've got one affected individual with cystic fibrosis, and that person is a female. In the third generation, so here we've got one more affected individual, and that person is then a male. In the fourth generation, we've got quite a few individuals who are affected. We've got 3 individuals who are affected, 2 of which are female and 1 of which is male. Now, this can tell you quite a bit about the genotypes of these people. So if, for example, we look at this person here, it might seem that they are unaffected by cystic fibrosis. However, because they've had a child here that's affected by cystic fibrosis, we know that they must be a carrier. Because the only way this person could have inherited cystic fibrosis is if they had 2 recessive copies of the allele. They're going to have obviously got one from this person here, but they need another one from a carrier in order to make sure that they are going to be cystic fibrosis affected. Equally, this person here who has cystic fibrosis is telling us quite a bit about their parents. Now, both their parents must be carriers of the disorder because neither of them are affected, but they've got a child who has it, and so this child must have inherited the recessive alleles. So now we're going to look at the last 3 topics in B2, and we're going to start with speciation. Now, speciation is the process by which new species occur, and a species is defined as a group of organisms that are able to interbreed to produce fertile offspring. So, first step in speciation is geographical isolation. And geographical isolation is where a population is separated by geography. So, for example, if you had an island and it's got a population of a particular animal on it, and the island splits one day all of a sudden, that population is also split, and the island is starting to move apart. And if it gets to a point where none of the individuals on one of the islands can move to the other island, then you've got geographical isolation. So if I do this with a little picture, we have an island, it's got individuals on the island, and then one day it just splits and it starts to move apart. And neither of those islands anymore can be reached by the other individuals on the other island. So now the next step is genetic variation. Now, genetic variation is the differences we've seen between individuals caused by genes. Now, on these 2 separate islands, as they start to float apart and as none of them can rejoin each other, the 2 populations will have different genes. Now, those different genes might mean that they're more or less suited to that environment, and it might mean they're more likely to survive and more likely to reproduce. The next step is natural selection. And this is just like in B1. So we have this genetic variation, which means we have individuals that are very different to each other. And then the next step is this genetic variation will lead to natural selection. So some species that are better adapted to survive on their particular island, or some individuals that are better adapted to survive on their island, will survive, and they will reproduce and pass on their genes to the next generation. So let's say we have these dots on this island, these individuals, and we'll say that this island gets really cold, and I'll symbolise this with some like shivering lines, I guess. Now, if this island gets really cold, let's say with some genetic variation, we've got a few individuals who carry a bit more insulation, a bit more blubber. These individuals are more likely to survive, while the other individuals are going to die out. Now, let's say on this island it was floating towards a really hot part of an ocean, and so any individual that has that genetic variation for a bit of blubber, well, actually they're going to die out on this island, and instead the other individuals who are a bit more adapted to having less blubber are going to end up surviving. Now, the last step is speciation. So over millions of years, this natural selection will lead to two populations arising that, although they came from the same island originally, have become so different through natural selection that they are no longer able to interbreed. So even if I took one of the individuals from this island and I shipped them over to the other island and tried to force them into breeding, it wouldn't be successful. They wouldn't be able to produce fertile offspring, and so at that point we know they are separate species. Now, if we think about extinction, so extinction occurs when a species completely dies out and there's no more of that species left. Now, extinction can happen because of many different reasons. So, for example, the environment can change and the species might not be able to adapt quick enough. It could occur because new predators arise. It could occur because new diseases arise and start to kill out a species. It could occur because of competitors. So, for example, if you think about grey and black squirrels in the UK, grey squirrels have almost completely outcompeted red squirrels and they're very close to extinction. There could be a catastrophic event. So this could be a one-off event that just seems to wipe out that particular species. So, for example, dinosaurs, we think, were wiped out by a catastrophic event, such as a meteor. And then finally, we need to know about fossil record. Now, fossil record is a good source of evidence for speciation and for natural selection. Now, the fossil record is a collection of all the fossils that we've discovered. Now, fossils can be created in several different ways. So if I talk about fossil formation for a second, fossils can be created through the hard parts of animals that don't decay. So if you've got bones, for example, inside your particular organism, the hard parts, those bones, won't decay as fast as its soft parts, and eventually you might form a fossil. Sometimes fossils are formed when there's an absence of the conditions needed for decay. So, for example, for decay you need it to be fairly warm, moist, and have lots of oxygen so that bacteria can decay your particular thing that's died. However, if the thing that's died falls into loads of ice, so I'm going to put e.g. here, ice, then it'll be frozen, and that'll form a completely different type of fossil. It'll form an ice fossil because the conditions needed for decay are absent. Now, ice fossils are really rare and actually incredibly useful if you ever find them, because you can even see things like the hair on the animal. But there are other ways that fossils can form. You can also have trace fossils. Now, trace fossils are things like footprints. So they're not fossils of the actual organism, but nevertheless they're kind of handy to have access to. And then the final thing is, some of your hard parts, like your bones, can also mineralise. So you might not have a straightforward fossil of the bones, but you do have a mineralised version of the bones. Now, fossil records it's really important. Like I said, it provides fantastic evidence for speciation because it shows you a record of how organisms have changed through time. The only problem with the fossil record is it's incomplete. And it's incomplete for a number of reasons. Number one, the conditions needed for fossilisation are rare. Number two, lots of early life forms were soft-bodied. So we have no record of them at all, which is really difficult when you're trying to prove things about how we formed from really early life forms like bacteria, etc. And then number three, geological activity can have destroyed fossils. So if you think about the rock cycle, in order to create fossils, you're building up layers of sediment on top of dead organisms. If you push those dead organisms deeper and deeper into the earth, they're going to come under extreme pressure and extreme heat, and actually they could just be destroyed. So the fossil record is really incomplete. However, the few fossils we do have show that speciation has occurred because we can see evidence of very similar organisms evolving and sharing similar traits. So that comes to the end of B2. Good luck.